My name's Joanne Berry. I'm an artist and I'm very interested in working with new media, digital technologies and industrial processes.
My name's Nick Holliday. I'm a group leader in the School of Biomedical Sciences. We're part of the University of Nottingham, so we're involved in teaching, but we also do quite a lot of research as well.
My name's Tim Self and I work in the Institute of Cell Signalling, and I'm a Chief Experimental Officer. My role is to develop and maintain the imaging technologies.
In essence, what we are all interested in is what we call molecular pharmacology, and really that's a posh term for trying to describe how drugs work. I guess what's special about our group and what we've really focused on is actually developing ways in which we look at how drugs work using imaging, and that's really how this collaboration with Jo has come about in the first place.
Tim Self, the Chief Imaging Officer, was very open about wanting to work with an artist.
I can't really remember what the email said, but it just piqued my interest.
The initial link was that microscopes use lasers, and I'm interested in lasers, and I'm interested in engineering, and I'm interested in technology. So all these things are in this department, and the imaging that comes out of this department is very interesting.
And the way in which I got involved was essentially to provide sort of the scientific focus.
I've been coming in a day a week and working here all day, looking at how cells signal.
We were quite clear, or Nick was quite clear, that the best way for it to do it was for her to actually follow all the processes and actually do them, the sort of thing that a student would do on the bench.
So we're doing things that are relatively straightforward, but also quite important in terms of what any scientist, any biological scientist might want to do.
I've had to learn about measurements, and the measurements are very different to measurements that I've ever encountered before, because I didn't do a lot of maths and I didn't do a lot of science.
We'll make up a number of different ligands or drugs, so we'll place it onto the microscope. The laser will excite the fluorescent dye. That light is then collected into a photomultiplier tube, and that produces a digital image, basically. You would excite it with one wavelength of light, and that excites that particular fluorescent molecule, and then it emits a colour. And the beauty of that is you can follow that colour within a cell, and you can put multiple different colours in a cell, so you can follow two different things happening with two different colours in real time on the screen.
Oh, there's something happening. It's very light.
There we go. The drug is there on the surface already. So that was really quick. That's much quicker than last week. Fantastic. Well, that's good.
What has been unique about this experience is that the imagery that I'm taking my work from has been imaged on the microscopes while I've been here. It's all about one specific project. I'm taking that imagery and manipulating it in different ways. So, for example, Z-stack of how a cell moves, I've been looking at different stills of that and producing layered drawings. I've also been manipulating the imagery through the computer software that is here, which is used specifically for microscopes, because you can do all sorts of fantastic things like stereo images, which are three-dimensional. You can colour the images in different ways. So I'm working a lot with the software that is here and then taking it back to my studio. A lot of the time when I was producing the drawings, I was actually hand drawing. I amalgamate the shapes together to create drawings that are like the scientific material that I was working on. Ultimately, each layer of drawing becomes a layer of perspex in a light box. I'm thinking about the cuts that are going to be made, that are going to work on top of each other when you put the different layers of coloured perspex together, and that's really, really important in the way I produce the drawings. When the drawings are finished, each layer is separated. The whole batch of the drawings are then sent to Implas, and they read them as separate CAD files. And then each layer is separately cut on the laser. Laser cutting is a great way of cutting out shapes and cutting out drawings in a very precise manner. It's also very, very, very fast, so that you can produce very detailed designs and they're cut out quickly so that the perspex doesn't burn or buckle. When they've all been laser cut, I bring them back to my studio. There's lots of unwrapping of the perspex, taking off the shapes that we don't need, and then putting the different layers of Perspex together. This isn't that simple because you try and layer different colours together and they don't look good, so you have to just keep swapping them and changing them carefully because Perspex scratches and it cracks, and I have had a few accidents with the sort of assemblage of the Perspex. Dusty. Right, you ready? It's been really fantastic working out of my comfort zone in a lab with two different people from very different backgrounds, and that has been really refreshing. It's been fascinating, and it's given me a deep appreciation of like sort of scientific endeavour. It's
been very useful seeing how she sits and works with the images themselves, because she works and she's doing different things to the sort of things that I do.
I think the imagery that I'm getting here and the way that I'm working is something that I want to expand and develop in the future, because it sort of highlighted a real interest in science.
You forget kind of, quite how amazing it is, because you kind of just get blasé about it. The fact that you're seeing very, very small proteins, at work in living cells. Jo comes in and she's saying, "Oh, isn't that amazing?" and things. You can suddenly think, well, this is what we're doing, and you can't quite believe that that's actually possible.